Good afternoon and welcome to the first of the webinar series on browsing genes and genomes with Ensembl. My name's Emily Perry and I'm the Ensembl Outreach Project Leader, and I'll be presenting the first of the webinars that you'll see as part of this series. Those of you who are actually at the webinar may be a bit confused because this is not exactly the same webinar as you were present at. Unfortunately, the recording of that failed, so I've had to re-record this. So what we're going to cover over the course of the next 7 weeks is what Ensembl is, what kind of data you can get from Ensembl, how to browse and navigate the Ensembl website, and finally, and I often think this is the most important one, where to go for help and documentation. So today's webinar will be an introduction to Ensembl with me. Next week you'll have Denise, who will talk to you about Ensembl genes. That'll be followed by Helen, who will take you through Biomart, a data export tool. And then Denise will be back to talk about variation data. Helen will tell you about comparing genes and genomes with Ensembl Compara. And then I'll talk about the Ensembl regulatory build, finding features that regulate genes. And then we'll finish on the 5th of May with Ben Moore talking about uploading your data to Ensembl and advanced ways to access Ensembl data. So the way each of these sessions is going to work is I will start by giving a presentation about what the data or the tool is and how we produce or process the data that goes into it. That will be followed by a demonstration where we show how to get the data or use the tool. And then there'll be exercises which are available on the Train Online course, which hopefully all of you have already found. For those of you in the webinars, since this is not live, unfortunately, this is possibly not so relevant, but if you're in the next webinars that come along, there'll be a chat box so you can ask questions during the webinar, and my colleagues will respond as the talk is going on. So this is the way it'll work in the next one, as well as the one we did yesterday, which didn't unfortunately record. There are also course exercises. So after each webinar there'll be exercises put up in the Train Online course. This video will take the place of the text that you can see in each section, and the links to the next pages will become exercises which will also appear in the page hierarchy on the left. And if you haven't done so already, you're welcome to join our Facebook group. There you can discuss the exercises with each other. And also you can ask our help as well. There's a link in the online course if you want to join the Facebook group. If you prefer not to work with Facebook, then you can email us on helpdesk@ensembl.org to ask for help with anything to do with the exercises. There are solutions available for them as well. So without further ado, I'll get on to talking about the basics and the introduction to Ensembl. So normally in a live workshop I would get other people to answer this question. Today I'll have to answer it myself. Why do we need genome browsers? Well, genomes are big, they contain lots and lots of data, and we're producing them at a fairly massive rate. And if you look at the kind of data that we're producing, it basically looks like this. It's completely meaningless. The only way this data means something is if we work with it and we process it and we do something with it, which is what we're trying to do in genome browsers. So these are the kind of 3 major genome browsers that are out there. There's ourselves, Ensembl, which I'm talking to you about today, but the other major ones are UCSC and NCBI Map Viewer, which are both very good browsers from the United States, and it really seems to be personal preference as to which one people seem to prefer. We have lots and lots of data in Ensembl. We have what we call gene builds for about 70 species. We take all of our genes and we compare them pairwise to each other, and we produce gene trees. We do what we call a regulatory build, which pulls in data from ENCODE, Roadmap Epigenomics, Blueprint, and predicts the locations of features such as enhancers and promoters. We have a variation display, which allows you you can look at known variation, and you can also use our VEP tool to analyse your own. We have various ways to display your own data, and we have various methods to export our data. So we have Biomart, which is a sort of point and click tool, and we also have APIs for programmatic access. We have a Perl API and a REST API. And one of the really cool things is that all of our code, all of our software is completely open source and free to use. Everything that I've just highlighted in red are things that are special and unique to us. So we make all of our own genes, we produce all the gene trees, we make the regulatory build, we made the VEP. We're the only browser which has got Biomart, we're the only browser with programmatic access, and we're the only browser which is completely open source. And you can access Ensembl data at various different scales. You can access things like genes on a one-by-one basis, you can access groups of genes, or you can access all the genes in an entire genome. And so for one-by-one access, you're largely looking at the main browser or the mobile site, but you can also look at groups or one-by-one with Biomart, the REST API and the VEP. The Perl API and the MySQL database are suitable for looking at every possible scale, and the FTP site is really for downloading whole genome data, so maybe every FASTA sequence in the database, something like that. We are used all over the world, so these are just our top 10 countries, but I only need to look at our list of registration for this course to see that we span far beyond the top 10 that you can see here. This is just based on our website statistics. And Ensembl covers vertebrates, so we try and cover a wide range of taxonomic space. We try and cover biologically important species such as model organisms, agricultural species, conservationally important species as well. And if you're eagle-eyed, you might have spotted that there are some non-vertebrates shown on this tree. We also have C. elegans, Drosophila melanogaster, and Saccharomyces cerevisiae in the database. These are in for a couple of reasons. One of them is historical, and the other reason is that they're actually really useful to have around when you're making gene trees. If you have a very unrelated species, you can use it to anchor the tree more effectively. If you work with non-vertebrates, you'd probably be interested in our sister site, which is ensemblgenomes.org. Ensembl Genomes has 5 mini-sites which cover bacteria, protists, fungi, metazoa and plants, and they all work in pretty much the same way as Ensembl. Throughout this course we'll find that there are a few exercises where we send you the way of one of these mini-sites, and we'll also point out any special features of the mini-sites that are important when you're looking at particular kinds of data. So the differences between Ensembl and Ensembl Genomes. Ensembl's been around since 2000. It was conceived as part of the Human Genomes Project, whereas Ensembl Genomes is a bit newer. We have the different species specialisation, but the major difference is the annotation. Ensembl annotation is all done in-house, so we annotate all our own genes, whereas Ensembl Genomes works with the scientific communities. And you access them by 2 different URLs. But I'll point out to you where you can jump from one to the other once you're in there. We also have a release cycle. The main benefit of the release cycle is it means everything is traceable. So just earlier this month we had Ensembl 84 coming out, and over the next few months what we'll find is various kinds of new data comes in, such as new assemblies, software updates, gene sets. Then for anything new we'll need to rerun our comparative genomics. We'll add in variation data, regulation data, as well as updates to any interfaces, and that will bring out the next release, which will be Ensembl 85. We're having a bit of a longer release than normal this time, so that will be out in July. And the main benefit is that everything becomes traceable. So when you find data in Ensembl, if you note down in your lab book which release you got it from, then if you go back to that data in 6 months' time, you can check not just the current version of the data, which may have changed, but also the older version of the data, and you'll know which version to look at. I just briefly, before I start showing you what's in Ensembl, want to go over genome assemblies, because this seems to be something that a lot of people aren't totally familiar with. So there's currently no technology that's available where you can start at one end of a chromosome and sequence all the way along to the other end. That would be wonderful, and I'm sure somebody's working on inventing it, but it's not out yet, which means that when we sequence genomes, we have to use other methods. So we have to cut the genomes up into small chunks, sequence the chunks, and put them back together. This inevitably leads to mistakes. There will be regions of the genome where we just haven't got sequence. There will be regions where we've put it back together, but we've put it back together in the wrong order. So this creates a genome assembly, which again we stamp a number on, or a name. And we can use this, so whenever we're working with genomes, it's important to note down which genome assembly we're working with, because if then somebody comes along, repairs a bunch of the mistakes, produces a new version of the genome assembly, you can know whether you're looking at the same one or a different one, and of course go back and try and find the one that you were looking at previously. Another thing to remember about genome assemblies is because we're cutting up genomes from real individuals, every piece of the genome is part of a real human or animal or plant or whatever it might be. So the human genome is made up of chunks of genome put together from a number of different people, whereas, and they've all got accession numbers, whereas other genomes, for example, the cat genome is all made up of one cat, which happens to be named Cinnamon. But the point is that every sort of piece of genome you're looking at, it is one individual sequence. So this can mean that if that individual has got a slightly unusual sequence at that region, that's still what happens to be in the genome. So we're going to do some hands-on. I'm now going to give you a demonstration of looking at the Ensembl homepage and how to find out information about species and genome assemblies in Ensembl. And then we're going to go and have a look at the human genome, and we're going to manipulate the view to see the data we're interested in. So we're going to look at a specific region. So I'm going to jump out of my presentation, and I've already opened up a tab with the Ensembl browser in. And there's a few things I'd like to point out on the home page for you. The first thing is this big blue bar that runs along the top. Now the blue bar is visible on every page you go into in Ensembl, which means you've always got a link back to the home page. You've always got a search box. You've always got links to help and documentation. We've also got a what's new in release 84, so if you're not sure which release you're looking at, if you go to this box over here, it will tell you what the current version is, and it will also tell you all the new things that came out. So when a new release comes out, you might want to go and have a look and see what's changed. If I scroll down to the bottom of the page, there's a little link here, and you'll see this on nearly every page, that says View in Archive Site. When I click on the View in Archive Site, it then gives me a list of previous versions of Ensembl that I can jump to. So if I've noted down in my lab book that I got my data from Ensembl 80, I can then go back and have a look at Ensembl 80 and see what data is actually available there. I'll just close that for now, I won't go and have a look. I've also got some links to some sort of common tasks that people like to do with Ensembl. I've got another search box which has the added benefit of a species selector, so you can search only a single species. And over here I've got more information about some of the genomes in Ensembl. So I'm going to go and click on View Full List of all Ensembl species. And this will now show me a full list of all of the 70 vertebrate species that we have. So we've got the images, the common names, the Latin names, taxon IDs, and also the assembly. You can obviously scroll down and search that way, but the quick way to find your favourite species is to type it into the Find a Species box over on the right hand side here. I can now jump into human. This is the species homepage, so I can see what's new for human in the most recent release, as well as some example regions, genes, transcripts, things like that. But the thing I'm going to visit is more information and statistics. And this gives me lots of information about the genome. So I've got a nice description here about when the genome was released, who produced it. I've got links to go and see the Genome Reference Consortium who made the human genome. I've got more information about gene annotation and also a detailed document I can read. Over on the right hand side I have tables of data, so I have things like the assembly version, the size of the genome, some dates as to when things were produced and released, and then things like counts of genes. Now you'll notice here it says GRCH38. That's the most recent version of the human genome, but we know that many people are still working with the previous version of the human genome, which is GRCH37. So if I just hop back to the home page, you'll see there's a link here to still using human GRCH37 and a link here as well. And these will take you to our dedicated site, which I have already opened up here, which is at grch37.ensembl.org. And this is a special dedicated website where you can access data from the older version of the Genome Assembly, and we keep it relatively up to date as well. Not quite as up to date as the main site, but there's still quite a lot there. So if you're working with the older version, this is where you want to go. I'd also briefly like to show you Ensembl Genomes. So this is at ensemblegenomes.org, and it looks pretty similar. We've got links out here and over here to the different branches of Ensembl Genomes. So we've got bacteria, protists, fungi, plants, metazoa, and a link back to the vertebrates. I'll just show you protists briefly, and you'll see that it's basically the same as we saw with the main site, except at the top it's purple, so we know which one we're in. But everything else works exactly the same. I'm just going to jump back and show you bacteria, because bacteria is special. And the reason bacteria is special is that we have about over 30,000 genomes, it's gone up, 30,000 genomes in Ensembl bacteria. And these are all different strains, different versions of many of the same species. Now obviously you can't really scroll down a list of 30,000 genomes, that would be horrendous. So instead we have this search for a genome option. So we have these two different search boxes: search for a gene, you put in your gene name, or search for a genome, which will then narrow you down to your genome of interest, and then once you get into that, you can just search within that genome. So if I type in my favourite bacterial genome, so Peptoclostridium, so it's already starting to find it because it's an auto, except that I mistyped. There we go, now it's working. Peptoclostridium difficile, it's already finding them because it auto-completes. And then I can jump to my favourite strain, Peptoclostridium difficile 630. And that takes me to a species homepage, just as I found with human. If I do a search now, it will, from this search box, it will only be searching this one particular genome. So this is a really good starting point, it really narrows you down. If I go into the information and statistics, you will see that I still have the statistics tables, but I've lost that paragraph because we don't have anybody whose job it is to sit and write 30,000 bits of information about the background. All of them are pulled in directly, all of the genomes are pulled in directly from the European Nucleotide Archive, as is shown here. So we don't do any full annotation on the bacterial species. So I'm going to go back to the main browser at ensembl.org, and now I'm going to search for a region and show you the region in detail view. So I'm just going to hop back to my presentation briefly, because we're going to use the search boxes now. Now the search boxes in Ensembl are really flexible. You can put pretty much anything into them. You can put gene names, Go terms, phenotypes. If you see a word or a phrase that you don't understand, you can put that in and it will search our help and documentation, things like our glossary and our FAQs as well. But you can also search for coordinates. Now when you search for coordinates, you don't actually want it to use the normal search because the normal search is using words. When you're searching for coordinates, you want it to know that they're coordinates and kind of bypass that. And the way to make sure it knows that they're coordinates is to use the correct format. Now the correct format for coordinates is to start with the chromosome number, followed by a colon, then the start coordinate, a dash, and then the end coordinate. Note that there are no spaces in this. So what I'm going to do is I'm going to select this, I'm going to copy it, and then I'm going to go back into the browser. Now I also have to tell it what species I'm looking for, otherwise there are loads of species which have got that coordinate, it doesn't really know what to do with it. There are two ways to do this. I can choose my favourite species from the drop down. So I could select human and just paste it in, or I can type human and paste in my coordinates. Now when I hit go or type return, it jumps me to my region of interest. So this is the region in detail view, and this is probably the most commonly used view in Ensembl, and there's a few things that I want to point out about it. The first thing is this little question mark here, and I recommend, if you're ever not sure what you're looking at in any page in Ensembl, you click on the question mark, because it will open up this pop-up window which gives you a description of the page you're looking at. So I've got diagrams, I've got text describing what I'm looking at, and in some of them, such as this one, I actually have a video. So if I hit play here, in a moment, Denise, who's doing the webinar next week, is going to appear.
Hello, I'm Denise from the Ensembl team, and I'm going to show you how to explore the region in detail view.
I'll just turn her off because I'm going to tell you all the things that she's about to tell you. And I'll just shut this pop-up. But they are really useful if you're ever not sure what it is you're working with. So the region in detail view that we're looking at is made up of three images. The image at the top is the chromosome. The image in the middle is a kind of overview. And the image at the bottom is a detailed view. So the chromosome at the top, this is our region of interest shown in this red box here. You can see the centromere, and you can see these sort of white, black and grey bands going across the chromosome. So this is really old-fashioned cytogenetic banding data, which is, a lot of people still use it as a kind of, this is my favourite, this region that's been identified. So we do keep it in there, even though obviously the more modern way to look at it is to use genome coordinates. We also have these sort of red highlighty regions at various points. Now these are haplotypes. These are highly variable regions of the genome that are represented as separate pieces of sequence because they are so very different between individuals. Now one of the easy ways I can navigate around, if I want to navigate around using the chromosome, I can do so by dragging out a box. I get given the option of marking the region or jumping to the region. I'll get rid of that, but that's one way to navigate around. You start to see a bit more detail when we go into this second view, which is kind of the overview. So this red box always means us, so this red box is us. And you can see a scale here shows us that the whole of this region we're looking at is a megabase wide. So in human, this will always be a megabase wide, but it will vary as to how wide we make this region, depending on the size of the genome in that species. So things like bacteria, this region will be very small because, of course, bacteria have very small genomes, they have very small genes, and they have very dense genes. So if you had it a megabase wide, you'd probably be able to see the whole genome, and it would just be really hard and really busy. So we can see our chromosome bands again. And we can also see this blue bar, which is labelled contigs. Now the contigs, I mentioned earlier about how the human genome was made up by chunks of sequence from different individuals. Each contig is one piece of sequence, so one single chunk. And the accession numbers tell us which piece it is. Some of them don't have an accession written on them. But they do have an accession. If I click on it, it won't work, I think, because I'm in the wrong tool. If I click on it, I get a pop-up that tells me what the accession of this particular contig is. So they do all have a number. We can also see genes. So these big coloured blocks here, so this gold block, this blue block here, these are all contigs. Genes even, not contigs. Try not to confuse everybody. Now, the labels of the genes are shown underneath them. So if I follow the leftmost point of a gene straight down, I'll hit the leftmost point of a label. So this big gold gene here is the SPATA5 gene. This gold gene here, if I follow it down, is the FGF2 gene. So if I follow down the left-hand side and go straight down, I'll meet the matching label. And I can navigate around this view as well using this drag or select option. So I'm currently in this double-headed arrow, which is the scroll option. If I click and drag anywhere in the view, it moves me around. It's like being in Google Maps. So I've got quite detailed control of where I'm dragging to. Now if I do this, the image at the bottom says, do I want to update the image? So I'm now looking at the region I've chosen here. Or do I want to reset the scrollable region back to where I was before? I'm going to put it back. If I change my tool option and I go for this dotted box instead, now whenever I put my mouse over the box, you'll see it turns into this dotted. I get the red dotted line. It stays as the big Mickey Mouse hand. But what I can do now is if I click and drag out a box, it offers me the option of marking the region or jumping to the region. So that's another way that I can navigate about. I will get rid of that box. And now I'll move down and look at the more detailed view at the bottom. So we've still got our contig, we've still got our chromosome bands, and we've still got our genes, but our genes are shown in much more detail. We've got exon and intron structure. And we've also split our genes into two different tracks. So tracks is a very genome browsery word. When we say tracks, what we mean is one particular kind of data that you can see plotted onto the view. So we would say that the genes are a track. We would say that this CCDS set, which are these green genes here, they're a track. And the contigs are a track. So it's a bit of jargon that we use quite regularly. So our genes are shown top and bottom. And the genes that are shown at the top are positively stranded, and the genes that are shown at the bottom are negatively stranded. We've also got some other positive and negative stranded features, so the cDNAs, the CCDS, we've got them at the top, but also at the bottom. Things like the chromosome bands, they're not stranded, so they're shown at the very top. And at the bottom, we've got regulatory features and variation data, which is also, and GC content, also data that is not stranded. So anything that's not stranded will be shown at the very, very top or the very, very bottom, whereas things that are stranded will be shown on either side of the contig, depending on what strand they're on. We have a legend at the bottom here telling us what all these colours in the display actually mean. Now this is a view that I can manipulate, I can change, I can make it show me different kinds of things depending on what I'm interested in. And I can do this either using this little cog or using the configure this page button. So if I click on configure this page, this takes me to a menu which shows all of the different tracks that are available for this view. And you can see there are a lot of tracks. For human we have thousands of tracks available. So you can see the name of the tracks. This is just showing us the ones that are currently switched on. And if I click on the info button that goes with each track, I can see a description of the track. So you might want to open up if you're not sure what a track might actually be. Because there are so many tracks, we've provided you with navigation down the left to help you to find the tracks that might be of interest to you. So I can click on variation if I'm interested in variation data, and then I could click on dbSNP variants, and you can see it gives me different options for how I want the track to be displayed. Now the easiest thing I find is to turn it on in a particular style, see if it gives you the data you need, and if it doesn't, change it. I'm going to turn it on in normal. And the other way that you can find tracks, so we found them quite easily using the categories, but if you're not sure what category something might be in, or you want to get to it quickly, you don't want to have to scroll through lists, you can use Find a Track, which is up here in the top right hand. So if I write a word like protein, that will then find me all of the tracks which have the word protein, either in the description, in which case the description will be shown, or in the name. So I'm going to turn on the proteins mammal from Uniprot, which I'm going to put in normal. I can now save and close the menu. We had a question in the live webinar, so I'll just let you know now about saving configuration. So you can click on this button, Save Configuration As. So if you've found a configuration that you use regularly, you can save it and it will save it to your session, which means that if you go back to the same computer, it will remember you using cookies, but you could also set up an account, and I'll just show you that when I've come out of the menu. And I can come out of the menu, I can either click on the tick, but I've put my mouse really giant so that you can all see it very well, which makes it very difficult for me to click on things. So I'm just going to click anywhere outside of the menu, which will also work. You can see it's loading. While that's loading, I'll just point out that if you want to set up an account with us, there's this login/register up at the top right, and it's completely free and nobody ever sends you spam emails. So, having filled the time by chatting, you'll see that I've now added some data. So I've added some proteins, which are stranded data. So we've got one at the top and one at the bottom. And I've added these dbSNP variants as well. Now, if you have added data, there is a quick way to change without going into the Configure This Page menu. So I can hover over the track name. You'll see it gives me the info again. I can change the style, so I can change it into labels. I can also turn it off as well if I wanted to. So if I turn that into labels, I will now see when it loads. We call this the spinning E of death. You can now see the labels that go with the protein, so these will be the UniProt IDs. I can move stuff around as well, so if I want to view these dbSNP variants alongside these genes here, you can see that when I put my mouse over the bar on the left, it turns into this double-headed arrow. I can now click and drag and move it so it's alongside other data. It makes it easier for me to compare two different kinds of data next to each other. Having found something that I think is interesting, I might want to share it with my colleagues, which is all very well if my colleague is the person on the desk next to me. I call them over, we take a look. But it's a bit more complicated if my colleague is somebody who works over on the other side of an ocean. Now, if we take a look at the URL shown here at the top, obviously what you're thinking is I'll just copy and paste that and I'll email them the page that I'm looking at. If we look at the URL, you can see it's telling us we're in Ensembl, we're in Human, we're at a location, and we're viewing it, and this is the location we're looking at. That's just telling us which database we're in. It doesn't tell us anything about all of the tracks that we've added, all of the manipulation that we've done. So if I was sending that to somebody, they would get to the same region, but they wouldn't see all the bits and bobs, and if you've done lots of manipulation, it might take them quite a while and quite a lot of description to get them into the same place that you're in. The solution to this is this Share This Page button over on the left-hand side. When I click on Share This Page, it generates a link, and this link includes all of the information that's included in this region view. So it's not just the location, but it's also all of the tracks I've added. And when Ben gives you the demo on uploading your own data to Ensembl, you'll see that when you try the share link, you get a little tick box with the option of do you want to include your own data as well. I can now put that in an email. I can send it to my colleague and they can be looking at the same thing that I'm looking at. So I will get rid of that. The last thing I want to show you, oh no, there's a couple of more things, sorry. I mentioned about the drag and select tool in the view above. You've also got it here. And I just want to point out, if I'm interested in this exon, I might zoom in. So I can do a jump to region. There is also this bar to zoom in if you prefer. But let's say I do a jump to region on this exon. I decide that I'm really interested in these variants here. So I can drag out a box around them. I can say mark region. I can then, we'll zoom out by dragging out a box here. So drag out a box here, jump to region. So when I did the mark region, you see it gives me this grey box. And the grey box is still there. So if I find something really interesting when I'm zoomed in, I can then zoom out and keep it marked and not lose track of where it was I was looking at. And then when I'm done with it, I can just get rid of it using this cross here. The last thing definitely this time the last thing. When you're going through some of the exercises, you'll find that you do add quite a lot of tracks into the region view, and it can get very, very busy when you add lots and lots of tracks. It becomes hard to see what it is you're trying to focus on. So what I recommend doing at the end of every exercise is going into the Configure This Page menu, scrolling down and hitting Reset Configuration, which will take you back to the original configuration state that it was in before. Okay, so I'll hop back so you can see it's back to how it was before. I'll hop back into my presentation and I've just got a few things to show you for kind of wrapping up today's webinar. So I've given you the introduction to Ensembl. Please do come back on the 31st of March where Denise will be talking about Ensembl Genes and also to our further webinars. So Denise will talk about the automated pipeline that we use to annotate genomic assemblies, as well as the manual annotation that we present alongside this. And she'll also take you through the Gene tab, which will show you the kind of range of data that you can get. Those of you who are tuning in live, please note the time. Our daylight savings time is taking place this weekend, so if you've already had your daylight savings time or you're not having a daylight saving time, please do take note that next week's webinar will be an hour earlier than this one, and all of them will also be at that particular time. For those of you who are in the live webinar, this was the point when I gave you the option of putting questions into the chat box. I will hop right over this now, but a lot of people did take advantage of this when we did this live. And the course exercises are available when you go into the online course at EBI Train Online. I will just briefly show you. So here we are on the Train Online course. As soon as I've finished making and processing this video, I will replace this text with the video there, and you can see that we've got the exercises shown you can have a go at, and also solutions to the exercises. So as well as asking for help on the Facebook group and emailing us, you can just have a look at some of the answers that we've got there. So yes, as I mentioned, please do have a look at the Facebook group and join if you would like to discuss the exercises with other people. There is absolutely tons of help and documentation available on Ensembl outside of this course. So many of you will have come in through EBI Train Online. There are tons of courses, not just on Ensembl, but on various other topics available there. We have tutorials. We have our YouTube and our UQ channel for short tutorials to take you through specific aspects, similar to the one that I showed you with Denise in earlier. And you can also email us. We are helpdesk@ensembl.org. And if you would like to follow us through social media, we do have Facebook, Twitter, and we have a blog. The blog's really interesting. If you're interested in kind of background to particular new kinds of data, then we will give you a bit of background to new stuff in the blog as well. We publish papers. If you do use our data, please do cite us. And this is the team as of June last year. So some of the people who will be giving the webinars and have been helping out with them are shown in this, as well as all the people who are producing all of the data and the interfaces you can see. Just to say thank you for everybody who takes part in the project, and also all of our funders as well. It would not be possible to deliver either the data and the website that we have, or the courses that we can do without all the funding that we have. So thank you very much for attending. I hope this was useful for you, and I hope we see more people back for the next set of webinars.